Shrews are one of few venomous mammals. The northern short-tailed shrew is the most venomous shrew, and yet, contrary to their broad diet, their venom is very simple. This begs the question, what is their venom composed of? How did the venom evolve? And what were the selective pressures? First, let's investigate its composition. A comprehensive molecular analysis was done by generating a de novo genome, 3 submaxillary gland transcriptomes, and sequencing the salivary proteome. Samples were taken from a female shrew labeled WH4 and the male shrew labeled WH5. A sample of the heart tissue was used to generate the genome using high molecular weight genomic DNA extraction. RNA was extracted from the submaxillary gland in order to generate the transcriptomes. The 3 de novo transcriptome assemblies were generated as follows: WH4 short read, WH5 short read, and WH5 short and long reads. By using both short and long read sequencing data, it can circumvent the overlaps in short read sequencing data that usually leads to erroneous chimeric contigs, while still accounting for the relatively higher rate of sequencing error associated with long read sequencing, therefore increasing the overall accuracy of the final results. After the transcriptomes were assembled, homology searches were performed against known sequencing data to identify protein domains and predict protein signal peptides and transmembrane domains. Transcripts that didn't possess signal peptides and did not match any known toxin in the database were filtered out. Signal peptides allow transcripts to travel out of the cell and potentially function as a toxin. The remaining transcripts were examined, and those in high abundance were considered potential toxins. This revealed 13 highly expressed genes from the ToxProf database. Using a combination of the new de novo genome assembly and the new de novo transcriptome assemblies, 3 novel KLK1 paralogs were identified. Of the 16 genes, 11 were undergoing positive selection, meaning these phenotypes were being favored over others by natural selection and increasing in frequency over time. Of these 16 genes, 5 were in high abundance in the salivary proteome. 2 additional salivary proteins were in high abundance in the proteome but not in the transcriptome. This makes for a total of 7 known or candidate toxins, namely BLTX, which is highly expressed in the submaxillary gland, undergoing positive selection and was the most abundant toxin protein in the saliva. This protein is lethal to mice as it decreases blood pressure. Next is a candidate toxin, KLK1-BL2, which was identified as having the same subfamily as BLTX. It is highly expressed in the submaxillary gland, relatively abundant in the salivary proteome, and it's undergoing positive selection. Sauricidin is a known toxin that is being highly expressed in the submaxillary gland, is present in the saliva, and is undergoing positive selection. It is known to immobilize mealworms, which is possibly used to store prey for later. Another candidate toxin, PA21B protein, was discovered in the submaxillary gland, is in high levels in the salivary proteome, and is undergoing positive selection. This may contribute to the paralytic effect of the shrew's venom. Hyaluronidase was found in the saliva and potentially aids in the diffusion of toxin components in the prey. TFPI2 is an anticoagulant that likely also helps in the diffusion of toxin. However, it is uncommon for this to be seen in mammalian saliva. Lastly, antileucoproteinase is a candidate toxin that is highly expressed in the submaxillary gland, the most abundant protein in the saliva, and is also undergoing positive selection. This is a serine inhibitor that regulates wound healing. However, it is commonly found in mammalian saliva, so its function in shrews is unclear. Based on 3D models of the 3 new KLK1 paralogs, it was determined that KLK1-BL2 was a candidate toxin due to its electrostatic similarities to BLTX, a known toxin. KLK1-BL2 has positively charged regulatory loops surrounding a negatively charged catalytic pocket, similar to BLTX, while their non-toxic counterparts have negative charges in both the pocket and the surrounding regulatory loops. 3 additional protein types were found in the saliva that potentially act as endogenous defense to protect the shrew from its own venom. 2 Cazal-type protease inhibitors were found. These act to inhibit BLTX. The anti-leukoproteinase protein is also thought to be a BLTX inhibitor. Lastly, endothelin-1 protein is a vasoconstrictor found in the submaxillary gland that counteracts the vasodilatory effects of BLTX. This is likely why they have a high tolerance to their own venom when it is extracted from their submaxillary gland. Now that we know that the venom is constituted of 7 lipoproteins and 3 possibly endogenous proteins, let's look at how these genes evolved. In order to investigate the evolutionary history of the KLK1 gene, a phylogenetic tree of the KLK1 gene from the northern short-tailed shrew and the common shrew was generated. The KLK1 gene subfamily follows a similar recruitment of venom genes via gene duplication as seen in other venomous animals. The reference genome of the northern short-tailed shrew contains 5 KLK1 paralogs, 1 known toxin, 1 likely toxin, 2 likely non-toxins, and 1 known non-toxin. The KLK1 BL1 gene is the likely ancestral gene to the KLK1 paralogs found in the northern short-tailed shrew, as it was present prior to them diverging. Finally, let's look at what possible selective pressures could have driven the evolution of venom in shrews. One common driver is a shift in feeding strategy. However, this is unlikely the case in venomous shrews, as their non-venomous counterparts also share their broad diet, and there was not a shift from active foraging to a sit-and-wait strategy. Another common driver is for defensive purposes. However, again, this is unlikely in the venomous shrews, as there is no aggressive behavior seen and there are no special venom delivery systems in place. One possible driver is that the venom evolved relatively recent. This is possible because the Blarina lineage is less than 10 million years old, which is evolutionarily young. Another possible driver is shifting to bigger prey. This is possible because the synergism of the toxins in their saliva suggests that vertebrates are their main prey target. Venom evolution in northern short-tailed shrews may have been driven by a dietary expansion towards larger prey. However, further functional assays of the candidate toxins are needed to complete the characterization of this venom system.